Our next speaker is Mandy Pritchard from the Cincinnati Zoo and Botanical Garden. She'll be speaking on American Burying Beetle husbandry, why dirt ain't doing it, and why Maggoty morsels are. She has always had a great fondness for the less lovable critters of the world. In the early 90s, she was the little kid running around barefoot, flipping rocks in the backyard, looking for snakes and bugs. Thanks to a high school zoology course that required students to make an insect collection, she was hooked. She went on to study zoology at Miami University and did an internship at the World of the Insect at the Cincinnati Zoo. In the fall of 2010, she was hired on full-time, and she intends to be at Cincinnati working with invertebrates, educating the public, and representing our spineless underdogs indefinitely. Her primary animal focus is with grasshoppers, katydids, and her personal favorite, the American burying beetle. Animals she's looking forward to seeing here in Arizona, any beetle will do, plus bombers. Welcome, Mandy.
All right, thank you all for being here today. I really appreciate it. I hope my voice holds out. I am losing it, so I got water with me, but we'll see how this goes. So I am the team leader at the World of the Insect, so increasingly I do more and more people stuff and less animal stuff. But one species that I do focus a lot on is the American Burying Beetle program, and that's what we're going to talk about today. So as an overview, I'm going to give you a brief history of this species at our zoo, and then we're going to talk about our current acquisition, breeding, and release methods. And then we'll get on to the meat and potatoes, which is this experiment that we ran over the last 12 months or so about housing and nutrition. So a couple of disclaimers here. Against my better judgment, I put videos in this presentation. So if they don't work, Winton has agreed to do interpretive dances for us. And also, I am a zookeeper by trade. So if you think that the way I'm presenting my data is weird or inaccurate, you're probably right, and you can come tell me about it later, okay? All right, so our zoo first acquired American burying beetles back in 1991, shortly after they were listed as endangered. In '93, we received a significant achievement award for breeding them. With no immediate plans to hold our own reintroductions like in our state or anything like that, interest waned. So we let that species phase out in the mid-90s. We reacquired them in '09. I was hired on in 2010. And then under the guidance of Randy Morgan, we established a release site at a nearby nature preserve called the Fernald Preserve. So we were holding reintroductions there for several years, and then COVID hit, and that kind of changed everything. At that point, we took a hard look at kind of our success at Fernald versus the success of our counterparts at the Wilds. It's another institution up in Cumberland, Ohio. And we realized that they were having much better success with their reintroductions there, so we decided to begin contributing our beetles to their reintroductions. So as for our current methods, we are fortunate enough to be able to acquire founders every single year from Nebraska, which is kind of amazing for a project like this. Specifically, we get them from the Sandhills region. Excuse me. We get them from the Sandhills region, which is kind of in the middle of that blue area in Nebraska there, near Bassett. So if you've never seen, this is an old range map for this species, but the green outline, of course, is their historical range, so basically everywhere east of the Rockies. And then in Ohio, you can see a few highlighted counties. Those are where reintroductions have been held in Ohio. So every summer, maybe you don't have to do a dance with me. Every summer we go out to, again, the Sandhills with a couple of keepers from our institution. We set some pitfall traps along the highways, and we come back the next morning and check them. The population out there is doing pretty well. Obviously, we're baiting these pitfall traps with carrion. By the way, if you're not familiar with this species. The population in that area is doing pretty darn well, too. This is all the beetles that we captured in just one night from one trap. So I think there were 56 in this particular one. And so we bring a certain number of founders back to Ohio with us. We split them between us and the wilds, and then we go into sort of our breeding cycle over the next year. So we will breed wild-caught beetles to one another once we get back to Ohio. And then in the fall, we'll start to trickle them into our existing population. In the spring, we'll breed the heck out of them so that we can produce like a really huge group to reintroduce in Ohio the following June. And then we'll hold a few back for breeding for subsequent generations. So if you're not familiar again with the species, they do require carrion as a larval food source. So they will bury a small carcass like a rat, maybe 200-300 grams. In the facility, we use 5-gallon buckets full of dirt. Give them a rat, beetles take care of the rest. So my phone did run out of battery during this, but you can see them kind of excavating the soil from under the rat and harumphing it into the hole. And in a matter of a few hours, sometimes all night, they will have it buried, sometimes to the bottom of the bucket. Very enthusiastic. So they lay eggs near the carcass, or the female does. Those eggs hatch, the larvae crawl down to the carcass, and they start munching, and the parents stick around and help take care of them. We check for larvae at about 14 days, and that's typically when you find these big, fat, juicy third instars. And then about So when we reintroduce these beetles, we do mark them before we release them. We'll use a surgical cautery tool and just kind of discolor one of the orange marks. That way, if we recapture it during pitfall trapping, we'll know that it was one we put out there versus one that was produced out there. And we do breeding reintroductions typically, right? So we'll provision each pair with a rat carcass, typically give them a little head start with a hole in the ground and let them do the rest. And sometimes in the fall, we also have like a huge surplus of extra beetles, and it's not the right time to provision them with a carcass. So we'll literally just plant them in the ground. So here you can see Kelly is encouraging this girl to go down the hole there, where she will stay until she decides to come up. Sometimes it's 3 minutes later, sometimes it's hopefully in the middle of the night. And the hope is that they'll stay at the site and overwinter, and then in the spring do what they're supposed to be doing, which is breeding. Okay, so on to the experiment that we ran over the last year. Because we're so fortunate and can get these founders every single year, we've noticed that the founders are really, really, really good at breeding, and the subsequent generations are not. So the average larval production for wild-caught parents over the last, 5 years, but this stops in 2022 because then our experiment starts, was like 23 larvae, and that drops to 12 for F1s. So it's like really frustrating. You go through this process, you're taking these animals from the wild, and then their offspring are just not being successful. The numbers for F2s are even worse, something like 25% success rate and like 6 larvae on average. It just gets worse and worse with every generation. And this is despite not inbreeding, which is like a huge faux pas for this species. So Amanda and I were wondering, how can we make this process better so that we can make better use out of the beetles that we're taking from Nebraska? So we thought, well, something's missing, right? Is it diet? Is it housing? What is it? So we decided kind of to look at both. By the way, if you've not met Amanda, she is here this week. She is one of our scholarship winners, and please say hi to her at some point. She has been totally instrumental in the ABB program over the last year, so we could not have done this without you. Anyway, so we decided first to take a look at nutrition. Sorry, I can't not put jokes in my presentations. So we did some preliminary trials with lots of different bugs, right? We're an insectarium, we had access to a few things. So we've offered waxworms in the past. They eat them, but they're not that crazy about them. Mealworms are their staple diet, as adults, by the way, not larvae, as adults. So we also offered roaches, earthworms, crickets, phoenix worms, chunks of pinky mice, and obviously staple mealworms. And all they were interested in was mealworms, which was really frustrating. Then we came across this paper that talks about an experiment that they ran with another couple species of Necrophorus, so same genus, where they fed them blowfly maggots, and the fecundity doubled. So the females were laying twice as many eggs, they were having twice as many successful offspring make it to adults, and then upon dissection later on in the experiment, they discovered that their ovaries were like twice as developed and twice the size. So we're like, well, there it is, and of course it makes sense. They're, flying to dead animals. What else are they going to eat at dead animals except for fly maggots? So Kelly Walker and I decided to catch a bunch of flies on our back porches, and our neighbors thought that we were totally insane because we just had like containers of rotten chicken and meat on our porch. And we captured lots of flies and we bred some flies. And we realized that the blowfly maggots are pretty small compared to the American burying beetle. So I think it was working in that experiment because the other two species are much smaller, but ABBs are like twice the size of other necrophorus species. So then we realized that Carolina Biologicals sells flesh flies, a similar species of necrophagous fly. So we just ordered some and then started rearing them in the facility. And once we had production figured out, we were able to feed them to the beetles and they devoured them. I love this video because you can see the mealworm in the background, but the beetle, like in broad daylight, hunted down this maggot in the front and is just chowing. So anyway, they definitely preferred maggots even over mealworms, which, if you remember, was their previous favorite. So then we decided to look at housing. And normally we would house them in these small plastic containers with moistened paper towels. And we thought, well, we can do better than that. So what else would a bug like to live in except for dirt? So we decided to mix up some topsoil with some leaf litter. We made it bioactive with springtails, and we thought, well, this would encourage them to behave like they might in the wild, where they can burrow into the ground and maybe even hunt for their prey a little more naturally. So once we made these decisions, we realized, well, we should set up a proper experiment and figure out what's actually going to work. So here we go. Sorry. Okay, so group A is our control group, right? They're housed on paper towels, fed mealworms only. Group B also housed on towels, but fed all the good stuff. Group C was housed on soil, but with mealworms only. And then Group D got all of the above, right? Between you and me, Group D was where my money was at. So when the offspring started popping up from our 2023 wild-caught breeding session, we evenly split some of the offspring from those buckets into each of the experiment groups, right? So a male and a female from every one of those 15 buckets went into each of the 4 groups. We also weighed the beetles every single week just to make sure that no group was falling behind. I mean, these are federally protected species. We didn't want to do anything bad. And then we fed them and housed them per their treatment group. And then later that fall, we bred them. We set up 3 sessions or cohorts, whatever you want to say. We were limited in the number that we could set up. I realized the sample size is really small. But at the end of the day, we still had to house and feed these beetles once they emerged. So we ended up setting 3 groups of 4 buckets at a time. So 1 bucket from each experiment group every single time. And then the results look something like this. So you can see Group B, hopefully, red and orange are showing up well enough on the screen there. But Group B pulls ahead with the the highest total larvae. Group A really, frankly, ticked me off with their total average larva because it was 42. And if you remember from that previous slide, all the other years combined, the average was 12. And this year it jumped to 42. So I don't know if they heard me and they were like, we're going to prove you wrong. But that was really frustrating. But that's how this goes, right? Anyway, so if you look at the total adults, though, that ended up emerging out of these buckets later on, Group B definitely pulls ahead. And then A and B kind of tied for average adults there. C and D did not do so well. Group A, however, was the only group that had a completely failed bucket. So they buried the carcass, but they did not produce any larvae. And then if you look at the survival rate, Group B really pulls ahead with that 81% compared to the mid-60s. So here's kind of just an overview of what I was saying. So Group A ticked a few boxes. Group B really ticked a bunch of boxes. Group C and D did terribly. Later in the experiment, they actually developed this weird small clustering mite problem that I want to talk to Bob about. And then they all died prematurely. So we're not going to talk about them anymore. So they were, yeah. Anyway, so when these offspring started popping up, we decided to continue the experiment, and so we just stuck with groups A and B, and we split the offspring pretty evenly amongst those groups, and then fed them and housed them in that manner, although housing was all paper towels again, until February when we began breeding for our release population. So in February, we set up 8 buckets from Group A and 10 from Group B. I realize those numbers are not even, which makes this graph a little weird to look at. But Group B just kind of ran away with it. So ignore the totals since we're dealing with 8 and 10. But if you look at the average, Group B had almost 3 times as many larvae and more than twice as many adults. And again, B is the ones that are getting maggots. Now, I will say here, though, Group B had a really, really, really low survival rate this generation. I think that it had more to do with soil quality. It was pretty common practice for us to reuse soil. We would freeze it between sessions, but I think something funky was in the soil because after we realized the beetles were not going to emerge, we went through it and we found all of these dead pupa that had been overgrown with like mold and fungus. That was not strictly in Group B, though. Group A also experienced some of that. It was just more from Group B had died. So Group A did have the higher survival rate, but even with that higher survival rate, Group B still kicked their butts. So, sorry, Group A. So we went on with Group B for an F3 breeding session. So once those beetles emerged, we fed all of them maggots, and then in May, we set up 4 buckets total. Not very many, I know, but we set up 4 buckets total, and all 4 of them were successful, which is completely unheard of, at least at our institution, for F3 beetles. And on top of that, their average larvae were 26, which is higher than the previous 5 years' average larvae for wild-caught beetles. So I think there's something to the maggots. I don't know. And then I know it's not really like apples to apples, but we did just do our 2024 reintroduction, and all of the beetles that we released from Cincinnati were fed maggots, and all of the beetles that the wilds had released were not fed maggots. And we were able to compare the two and how they did in the reintroduction, and the maggot group outperformed the non-maggot group yet again. So in short, we will be feeding our beetles maggots from now on. If you're interested in raising flesh flies, be warned, it is very stinky, but I am happy to share my husbandry protocol with you. We raise them on ground beef in 1-gallon jars, and my coworkers love it. Yeah, so, and also the adults make excellent prey items for things like trichonephila and mantids, and we even freeze them and feed them out to our aquatic invertebrates. So that is all I have for you.
I think we have time for a question.
Hi, kind of a simple question, but I noticed when you guys were planting the female individual and encouraged her to go into the substrate, was there like a yellow neon dot to identify her? What is that? Like, how'd you guys do that?
Yeah, that is a bee tag that you would use to tag a queen bee. One of the years we decided to adhere bee tags to all of the beetles before we release them. With mixed results, the beetles tend to rub them off pretty quickly. We adhered them with super glue. So there were mixed results. It definitely wasn't foolproof. At least in the lab, probably 50% of them fell off. So.
We have one final question from Kayla Garcia online. And she's asking, have you ever compared the nutritional composition of fly larvae compared to waxworms?
Fly larvae to waxworms, no, I haven't. We did send the fly larvae out for nutritional analysis. So if anyone's interested in that, I'm happy to share it with you. And if anyone wants to interpret that with me, I would really appreciate it. We took a look at it with our vet staff and our nutritionist, and they told me, I think, some very interesting things, but I think they're coming from like a very vertebrate-centric point of view. And so somebody that knows insect nutrition should please come talk to me. But no, I haven't directly compared them to waxworms.